Okay, this is biology section 3. We're going to go from the very beginning to the very end. So the first point in the syllabus is a definition. So as per usual, for definitions, all you do is you learn the definition and try to break it down. So asexual reproduction is the process resulting in the production of genetically identical offspring from one parent. So key points, one parent. That was asexual, so sorry. One parent and genetically identical. Okay? If you go back, we need to discuss the advantages and the disadvantages for sexual and asexual. So what we'll do is we'll go through these two first and then discuss the advantages and disadvantages. Now, your definition for sexual reproduction is the process involving the fusion of haploid nuclei to form a diploid zygote and the production of genetically dissimilar offspring. So what that means is you have two parents, in this situation a blue and a red, and they produce haploid cells, which are half of what they are. Okay, so they produce these haploid cells, and each of these halves meet up to make a new diploid cell, and that diploid cell is genetically similar but genetically dissimilar to the parents. Okay, so that's sexual. And asexual, like we said, identical parents splitting to become parents splitting to become two identical offspring. So the red splits and becomes two identical reds, the blue splits and becomes two identical blues. The pros and cons of sexual reproduction. The pros are your children are not the same, so that minimizes competition. Great example, if everyone likes Skittles in your family, then there's a fight over who gets the Skittles. But if one child likes Skittles, one child likes Reese's, one child likes M&Ms, there's less of a fight over each one because there's enough for everybody. The downside to sexual reproduction, it is slow, very slow. You need to find a mate, so if you live in the vast African savanna and it takes you 3 days to travel to your mate, you will probably die on the process. Sex reproduction produces very few offspring, and a downside is one of the upsides as well. All your children are not the same. If you live in the land of Skittles and your children, some eat Skittles and some eat M&Ms, the ones who eat M&Ms are probably not going to make it. The pros of asexual reproduction, so that's just the cell splitting to make identical offspring or daughters. There are many. There's no need for a mate. It's super quick, and all your kids are the same. And just like in the sexual reproduction, the downside to that is all your kids are the same, so we introduce more competition. So key points from those two objectives. The term genetically identical, the term haploid, diploid, zygote, and fusion. They are straight. Context clues could help you figure out what they are, but haploid means half and diploid means double. So when a cell has half the amount of chromosomes it needs, it's haploid. Just a point to note, we'll get into it later, haploid cells are called gametes or sex cells. So we get into reproduction in plants first. Identify and draw. So very basic. You need to be able to draw a picture or get a picture of a typical flower and identify the parts on it that you get from the syllabus. So petals, stamens, sepals, etc., etc., etc. So let's start with the carpel, which is the female part. So I put it in pink. The landing pad is called your stigma, and then you have a tube from the stigma all the way to the ovary that is called your style, and then you have the ovary which houses the ovules, and that is at the bottom of the style. And that all of those together make the female sex organ, so to speak, and that is your carpel. The male part is called the stamen, easy to remember, has the word men in it, and it has two parts, an anther, and the filament. Okay, so this is insect pollinated flower, you can tell by the large petals. If we get to a wind pollinated flower, slightly different in approach, but the same idea. Your male part, anther and your filament, your anther is much bigger, your filament is much longer, and of course that's together called the stamen. And then the female part, you have your stigma, your style, and your ovary again. But this time, instead of your stigma being a large landing pad for insects, it's these little feathery pieces that catch pollen that floats through the air, because the pollen is the male sex cell. Okay. You need to be able to state the functions of sepals, petals, anthers, stigmas, and ovaries. Straight from a BBC bite-sized site. There's a little table for you to copy down, and you just need to create a table for that one. It says use a hand lens to identify and describe the anther stigmas of one locally available named wind pollinated flower and examine the pollen grains under a microscope. Whenever it says locally available, just throw the word Bermuda in. So Bermuda grass and draw whatever was in the textbook. There's no point in going crazy over trying to figure out and find these things. If they want to wind pollinated plants are normally grasses, Bermuda grass, and then you draw the basic one from the textbook. The point in this syllabus where it says you should be able to apply your understanding of flowers to unfamiliar flowers. If you are shown a flower that you've never seen before, you should be able to identify anthers, stigmas, carpels, etc., etc. For wind pollinated, the anthers hanging out and the female part looks like a feather. You just move on with that. Next, you need to define pollination. So pollination is the transfer of pollen grains from the male part of the plant to the female part of the plant, basically the stigma. So that's just the same as how humans, quote-unquote, pollinate, so to speak. And the agents of pollination, so straight from the syllabus, name the agents of pollination. Agents means the mechanisms by which pollination is encouraged or enhanced. So you have wind, blowing pollen from one flower to another, insects who move pollen from one flower to another, and humans who also purposely move pollen from one flower to another. So here's another quote-unquote table of what the sepals, petals, anthers, stigmas, and ovaries do. And now you have to compare the differences between a wind-pollinated flower and an insect-pollinated flower. And it's straight in the syllabus. Compare the different structural adaptations of insect and wind. So the big difference, insect-pollinated flowers are designed to attract the insects, so they're big and bright. They make nectar that is sweet. The filaments are short so that they're inside of the flower body where the insect wants to go and it brushes against it. The pollen is small and sticky, as is the stigma, also sticky. Wind has little to no petals. They do not make nectar. Their filaments are long and hang outside of the flower. They produce lots of pollen that blows in the wind, and the large feathery stigma catch that floating pollen around. So next you have to investigate and describe the structure of a non-endospermic seed in terms of the embryo and testa protected by the fruit. So here we have your basic seed. The outside of it is called the testa. I peel that outside off. What I have is an embryo or baby plant, and there are two different parts to it. The radicle, which is what would eventually be the root, and the plumule, which is what would eventually be the shoot. The micropyle is a small hole in the outside of the seed that allows water to get in and actually gives a space for the radicle and the plumule to shoot out. There is a bunch of food that surrounds the embryo called a cotyledon, and once water and temperature are right, that cotyledon is very starchy. It starts to break down due to enzymes into simple sugars and amino acids, and that will feed the embryo, allowing it to grow even without sunlight. Now there are different ways in which seeds get around. And seeds is different than pollination. Make sure you get the differences down. Seed movement and pollen movement are two completely different things, even though they might have the same mechanisms. So seeds travel by wind, animals, water, basic bursting, and humans. Okay, and there are some examples. If they want a local example, just put the word Bermuda first. Now, why do seeds have multiple ways of dispersing? It's because plants want to colonize multiple places. They want to move on and spread their seeds all over the place. Colonize, good joke. All right, now let's take this tree for example. If it drops its seeds right below its canopy, the sun and the rain will not get to the seed, so that little tree will die. But if it found a way to get its seed to go outside of its canopy, the sun and the rain can reach it, so the tree will grow. So trees have developed ways to get their seeds as far away from them as possible, or just far away enough for them to not be covered and in competition with mommy. Okay? So we get into fruit production and pollination, the depth look. So again, pollination is when you get the pollen to go from filament to another stigma. So let's look at insect pollinated. When the little bumblebee goes to try and get some nectar, the anther touches the bee and pollen grains are distributed onto it. When it flies to another plant, the stigma touches the bee and accidentally picks up some of these pollen grains. So it's when pollen grains move from one to the other. Trees can either pollinate themselves or find ways to not pollinate themselves based on when the anther or the pollen grains ripen and the stigma is ripened. Okay? Once pollen does hit a stigma, it germinates and it grows a pollen tube that permeates through the style and into the ovary, and then the nucleus from the pollen grain floats down this tube and it fertilizes an ovule. And that fertilized ovule starts to divide by mitosis, and you get a seed to form. The ovary wall sometimes will swell, and that becomes a fruit. And that's how you make fruits. So we're going to steal someone else's notes on self-pollination versus cross-pollination. And let's just take a look at this picture here. Self-pollination is when the pollen of a flower goes to the the stigma on the same plant. And then cross-pollination is when it goes from one plant to another. Okay, so you have a definition here. So many definitions, and then the pros and cons, all right? Pros of self-pollination: there's a better chance that you'll pollinate because it's your brother. You increase the number of successful pollinations. The downside is there's less variation, so just like sexual versus asexual, everyone's going to be pretty much the same. Cross-pollination: less successful, less chance of fertilization, but you'll increase the amount of variation between you and your children. And by you, I mean plant. So we're talking to a tree right now. So just some points to remember. The male gamete, or the nucleus of that sex cell, is inside of the pollen grain. The pollen grain is just the delivery mechanism. Okay? Part 2, after the fertilization of the ovule happens, you have mitosis occurring, where that cell keeps splitting and splitting and splitting, and then it develops, it turns into a zygote, and that zygote turns into an embryo, and then the ovule turns into a seed. Alright, so the ovule is the outside, that turns into the seed, and the embryo is inside of the seed. The ovary wall swells, and that becomes fruit, or the flesh of a fruit. So you might have to rewind that and play that a few times. Make sure you know those steps. Pollen grain lands, the pollen tube develops, the nucleus floats down this pollen tube and fertilizes the ovule. That starts to split by mitosis to become a zygote, continues to split to become an embryo, and then the entire ovule turns into a hard seed. Alright? So that's seed formation and fruit formation. So we move in now to the male reproductive system and the female reproductive system, so we get into humans. For the male reproductive system, you have to be able to identify in diagrams testes, scrotum, sperm ducts, prostate gland, urethra, penis, and state the functions. So we have our, oh, that's not the male. That's the male, and we need to label these, so let's just put some labels up. Okay, so there we have some labels. The bladder, the sperm duct, the urethra, the scrotum, the prostate, the testes, and then I didn't label the penis. You need to be able to say what each one does, so there's a little, you could turn that into a table if you wanted to, for the male parts. And then let's move on to the female parts. So this list is a little shorter. You just need the uterus, the vagina, the cervix, the ovaries, and the oviduct. And then again, you have to say what each one does. Oops. And we're going to steal this little girl's notes. Bang! Okay. The next topic is the menstrual cycle, which is all about preparing the nest for the egg. Okay? So the uterus is our quote-unquote nest, and over the course of a month, the nest builds up, getting nice and ready for implantation. If implantation does not happen, it all gets washed away like the plague, and then it starts again. Now what controls this cycle from happening are a bunch of different hormones. So your first hormone is from the pituitary gland, and it is called follicle stimulating hormone, or FSH. Alright? And that starts the cycle by stimulating the follicle to develop. So as the follicle develops, the egg cell starts to divide, and it gets larger and larger and larger. That egg cell and the cells inside start to secrete estrogen, which is a hormone right there. So if FSH causes the follicle to start to divide, as that follicle starts to divide, it starts to give off estrogen. Now estrogen encourages the lining to thicken. So estrogen says, let's get the nest ready. And as the estrogen goes up, the lining of the uterus starts to thicken as well. As the estrogen increases, this tells the FSH to stop. So the highest point in estrogen normally is the highest point in FSH as well, because once estrogen gets to a high point, FSH starts to drop off. So the follicle stops being stimulated. As FSH drops, LH gets to its highest point, and that's called luteinizing hormone. And luteinizing hormone tells the follicle to release the egg. Now, the little house that was outside of the egg, or the follicle, turns into what's called the yellow body, or the corpus luteum, and that gives off progesterone. And progesterone helps thicken and maintain the uterus lining, and there you have it. If pregnancy occurs, you have a nice big fluffy nest for implantation. If it does not, the lining gets to its maximum thickness, and then it all gets sloughed away. So what I would suggest you do is make a table. Well, you would have to turn this picture into a table. Which hormones are secreted by which functions or which bodies, and what it actually does. Yeah, that's as much as I'm going into. Now, if you look straight in the syllabus, it says describe the menstrual cycle in terms of changes in the uterus and ovaries. So I'm 50-50 on if you need to know what hormones are made. You just need to know what's happening where. Uterus gets thick, builds up, and then it all sloughs away. We call that menstruation. In the ovaries, your follicle develops and grows. Ovulation is when it releases the egg, and then the corpus luteum starts to degenerate. So let's assume that instead of menstruation we get fertilization. there's a common misconception about what fertilization is. It is not when the sperm meets the egg. It's primary school. It's when the nucleus inside of the sperm cell fuses with the nucleus inside of the egg cell. That is fertilization. So step 1, step 2, step 3 is not fertilization. Only step 4, when that genetic material meets this genetic material, and that's when you get fertilization taking place. Alright, so once fertilization happens, so the nuclei form and fuse, you start to get mitotic division. So the cells start to divide, and you go from just 1 cell to 2 cells, 2 to 4, and we get the zygote, which is this stage, becoming a blastocyst. And then the blastocyst meets with the uterine lining and starts to implant. Okay, so you go from 2 different meiotic sex cells or haploid sex cells. They meet, they start to divide, and they become a zygote. And then the zygote keeps dividing until it becomes a blastocyst, and that will eventually become an embryo. And then we get our next steps. Some points to note about sex cells or gametes. Male sex cells or gametes are small, they swim, and millions are produced. Females, on the other end, are much larger. They do not move on their own, so that should be changed. They float, and they are matured or released once a month. So you need to outline birds and bees, and there you go. Oops. So insert A into B, mix together, and you're done. So there you go, an outline, not getting into that. So next you have early development of the zygote, simply in terms of the formation of a ball of cells that becomes implanted in the wall of the uterus. So there you go. The fetus starts to develop and grow. Oops, wrong one. And the way it gets so the blastula develops in an embryo, and then it attaches to the wall via what's called a placenta. Now the placenta is how the fetus gets its nutrients and its oxygen, and it's literally just a bunch of pins that stick into the mother's uterus that allow diffusion to take place. Okay, so we need to talk about amniotic sac and amniotic fluid, and we'll go back to the PowerPoint. So the amniotic sac, or the amnion, they protect the fetus from impact and mechanical damage. Imagine if you had an egg inside of a beach ball filled with water. You probably couldn't break that egg if you tried. Now, your placenta is just pressed up really close to the uterine lining, and the umbilical cord connects the baby to the placenta. So oxygen and nutrients can get to the baby via diffusion. The blood never actually mixes. It's kind of like a sheet of glass between two people. They can hear each other, they can see each other, they can even feel the warmth between each other, but they're not touching. Same thing happens with the placenta. Gets really close and allows diffusion. Remember, diffusion goes from where there's a high concentration to where there's a low concentration. Now, in this diffusion process, everything diffuses across that the baby needs, and the things that the baby does not need diffuses across to the mother. So urea, poisons, everything. So we probably shouldn't even say what the baby needs. Even things that the baby does not need will go straight across. So then we go into the process of birth. Starts with labor, finishes it up with cutting of the umbilical cord. There's a nice little flow chart for you. Now after the baby is born, the mother has to maintain a semi-decent diet, and they call this antenatal care. Sounds weird. So they need to have sufficient calcium, iron, carbohydrates, and proteins. Calcium for bones, iron to keep the hemoglobin in the blood high, carbohydrates for energy, and proteins for amino acids and growth. If she decides to have the worst diet in the world while pregnant or after the baby is born, there will be some problems because she has to breastfeed. The upsides and the downsides to breastfeeding, there's a little list. Please note all the downsides are easier to overcome. Okay, so I don't even think that they're a downside. So there's your list, and if I can find my mouse, I will pause. I feel like we're going backwards, but then we get into the hormones associated with puberty. So the two main ones for girls and for boys. In girls it's estrogen, and in boys it's testosterone. So we have a little table of where they're secreted from and who it makes changes on. 50-50 on that, if that one's true. And then the secondary characteristics, how do those changes affect males and females? Please note there is a point in the syllabus where they specify you need to know the differences in relation to pregnancy. And it's very simple. In pregnancy, you add a hormone, you add progesterone, and it comes from the placenta. Okay? Methods of birth control, they break down into natural, chemical, and mechanical. Technically surgical and mechanical should be the same, but let's make it 4. Natural, chemical, mechanical, and surgical. So for males and for females, it's split up, males on the left, females on the right. Males get a vasectomy where you cut and tie the sperm duct. Females get a tubectomy where you cut or tie the oviducts. Mechanical, condom, female condom, diaphragm, and IUD. There's your information. Table's broken down for you already. Chemical is either the pill or spermicide. This one's backwards. The male should be on this side. Is that backwards on the other one too? No. Oops. And then natural, yeah. This is probably the only one that works in that. Okay, so make a little table. Like I said before, tables are biologist's best friend. Into artificial insemination. If you are having trouble getting the pregnant, you can use some artificial insemination or some help from the doctor. The first one is just basic drugs to, maybe it's this one. Nope. Get some drugs to help stimulate your ovaries to produce more eggs. All right, so those are like fertility drugs. If fertility drugs do not work, you can, if it's the male's problem or maybe his swimmers aren't swimming, you just use this little syringe, and this is the insemination part, and you deliver the sperm exactly where they need to go. If the problem is that the female has blocked oviducts or damaged oviducts, you can just remove an egg and add a sperm to it in a test tube, and then once you get your zygote, splitting and forming, you then just deposit that in the nest. Of course, for this one as well, you're going to need to have some progesterone and some estrogen in there to build up that lining. The fertility drug that is used to make the ovaries release the egg is going to be FSH, just like in your menstrual cycle, and the drugs needed to build up the lining would be estrogen and progesterone. So instead of letting your body make it, you make it yourself. Okay, so that's I don't know when the last time I was recording was, so I have to double check that. Social implications of artificial insemination. If you use a donor, you don't have the daddy. A big part of it is feelings. Females, if it's their reproductive system that's faulty, they feel like they're at fault. Males, the opposite way around. One big one is when you use fertility drugs, you increase the chances of pregnancy and multiple pregnancies. So instead of having one kid, you have 5 or 6 or 7. So those are your social implications. Hey, have fun with that one. STDs, the big ones for us are gonorrhea and the HIV. So we go through the HIV next, we go through gonorrhea first. You need to know symptoms, signs, and effects and treatment of gonorrhea. Symptoms, signs, effects, and treatments, and a nice little table for you. So there's your table. Now for HIV, this is me being artistic. 7 ways to get HIV. Just turn your head if you can't see it. See, that says HIV, but it's also the ways. Yes, I know. What does HIV do? Well, basically it's how your immune system works. There you go. Normally your lymphocyte, so what those slides just showed, your lymphocytes would create antibodies which either destroy or label pathogens. Okay? Now, the way they know to do this is they have these things called helper T cells. And what the helper T cell does is he finds the germs and he tells the lymphocytes. And he says, help, help, help. And the lymphocyte makes the antibodies that either destroy or clump together or label the germ. Now what HIV does is it destroys the T cells. So now you have no one to tell the lymphocyte to make antibodies. So the germs don't get destroyed and eventually you die of some immune disease. Okay, so you don't want to get the HIV. So how do we prevent HIV? Well, if you look at these previous slides, just don't do what these ones say. Oopsies. Don't share needles, don't have unprotected sex, and make sure that your blood transfusions are clean. Because you're being born, you can't really control the other ones. Okay? So growth versus development, you'll find that a lot when it has to do with seeds and the fetus forming. So growth is just a permanent increase in size, while development is an increase in complexity. What you'll find is as organisms grow, they need cells to be more specialized. So if you're just 4 cells, you don't need cells to have any specific jobs, just do whatever you're doing. But if you turn to 60 billion, then somebody needs to be responsible for sight, someone needs to be responsible for creating enzymes, someone needs to be responsible for moving oxygen, etc., etc., etc. So that's growth versus complexity. And straight from the syllabus, you have definitions of the two. Investigate and state the environmental conditions for germination of seeds. Seeds need water, oxygen, and a decent temperature. The fact that they need oxygen, this lets you know that there is respiration taking place. You can drown seeds if they need oxygen to respire, and you don't provide them with that, they will not grow. The suitable temperature, that it lets you know that it has something to do with enzymes. Enzymes work best at warm temperatures, so suitable temperatures and respiration are what take place in the germination of seeds, and the water activates these enzymes. Okay, so growth versus development, and we're going to talk about meiosis and mitosis real quick. Mitosis is just regular cell division. One cell turns into 2 cells, and they are both identical. Alright? Meiosis is where the cells are not identical, they're cut in half, and they stay in half. But the way they're cut in half isn't directly, you kind of just break them into a lot of pieces and take half and mix it back together. So this is how you create sex cells, alright? So meiosis is reproduction by division through which the chromosome number becomes half. So if you have 46 pieces of Lego together, what you do is you smash it all into bits, and then you put 23 pieces together on one side and 23 on the other. Now you have 2 halves of 23, which look nothing like the original 46 that were stuck together. Okay, that's meiosis. Mitosis is where you put the 46 pieces in the microwave, you microwave it, and out pops 2 sets of 46. It's a magic microwave in this situation, guys. Just some more definitions, and they come straight from syllabus as well. Haploid and diploid. Haploid is a nucleus containing a single set of unpaired chromosomes, so they only have 23, there are no pairs. And these are your sex cells: sperm, egg, pollen, ovule. Please note it's the nucleus inside of these guys that has the chromosomes, chapter 1. And then your diploid nucleus, 2 sets of chromosomes, so they have 46. Alright? Now we get into how describe the inheritance of sex in humans, X and Y, how boys and girls are made. Alright? So a female has 100% of her chromosomes in the sex cell is X. So every egg she makes is an X. Now a male 50% of the sperm he makes are Y, and 50% of the sperm he makes are X. So when a Y sperm fertilizes the egg, XY makes a boy. When the X sperm hits the egg, XX makes a girl. So the people who claim that drinking orange juice helps you have a boy, no. That is determined the second fertilization takes place. If it's a Y chromosome that hits the X egg, then it's a boy. If it's an X chromosome that hits the X egg, then it's a girl. That's it. And that is how you get diploid cells, you know? Half of your set comes from your father, and half of your set comes from your mother. Diploid. A point to note, meiosis and mitosis must be spelled correctly in order to get the marks on the exam. Inheritance. The inheritance can be tricky, so I figured out a way to explain it using an analogy that hopefully you get. Okay? So inheritance is the transmission of genetic information from one generation to the next. So it's you giving a bunch of information from you to the next generation. Alright? So inside of your cells, we have a nucleus, and that is packed with chromosomes. As I start to unpack my chromosomes, I am able to see that it is made out of many genes. And each of these genes are a recipe for a protein. So that's the basics of inheritance. So again, nucleus of a cell. Inside we find a chromosome that is just a bunch of tightly wound DNA. We unwrap that DNA a bit and we can see each gene, and every gene is a code for a protein. Now, a chromosome is a thread of DNA made up of a string of genes. We just said that one. So what we're going to say is that the chromosome in this situation is a suitcase. And inside the suitcase, you're going to find genes. And genes is a length of DNA that codes for a specific protein. In our situation, our genes always code for an outfit. And your outfit is made out of tops, bottoms, and footwear. Okay? Now, if there are different types of genes, we call it an allele. So any two or more of an alternative forms of a gene. Alright? So these are all alleles. They are all genes, but they're different types of the same one. Each gene codes for an outfit, and this is outfit 1, outfit 2, and outfit 3. So those are your alleles. Okay? Now, then we get into genotype. The genotype is the genetic makeup of the organism in terms of all the alleles present. So, the genotype is everything that you have on. So everything about this outfit. The tops, the bottoms, the footwear, everything. The phenotype is only the things that you can see. So that is due to the environment. So if we look at this genotype, because it's cold outside, we wear the hoodie. But in this phenotype, all you see is the hoodie. We know that we have on underpants, but you can't see the underpants in the phenotype because it's hidden. The genotype, you know that the underpants are involved because you get to see all the genes. So imagine your grandma has green eyes. You have the genotype for green eyes. Do you have green eyes? No. But you still have that gene somewhere in you. Okay? So the phenotype is just what you can see. Imagine if you had genes to be tall in your family, but you decided not to ever drink your milk or eat your vegetables, so now you're a hobbit. Well, that's your phenotype. Your genotype is still tall. You can have kids twice your size. Now for genetics, we express our genes as letters sometimes. Alright, so homozygous is having two, that should be the word two properly, having two identical alleles of a particular gene. Two identical homozygous individuals that breed will be pure breeding. So that's rocket science right now. I'll explain it. So let's see what we got next. A homozygous allele would say that you got two together. You always pair them up. Well, you don't always, but you pair them up. No matter what, if these two outfits were to mix and match, they would always be the exact same outfit. Alright? Heterozygous is having two different alleles of a particular gene. So the gene is for outfit, and two alleles are, red hoodie and denims, blue and capris. Now when I mix these, it's going to come out different every time. But we have something called dominance and recessive. The dominant allele is the one that is always expressed if present, and the recessive allele is the one that is only expressed if there's no dominant. So in this situation, in all honesty, the dominant allele would be the hoodie, because that one will always show if you're wearing it. Alright? That's not the best example. So we show these with big letters and little letters. Now they always need to be in pairs in this level, because we're doing monohybrid inheritance. And the first thing you need to show is the phenotype. So we're going to talk about tall and short. Now, for tall, capital T means tall, lowercase t means short. You don't switch the letters, okay? So let's look at the parents. Both parents are tall, but they're heterozygous. And the capital letter is also the dominant one, all right? So as long as there is a capital T, this kid is going to be tall, and if there are two capital T's, then he's definitely tall. So what we do is we break down our genotype. We break it down into sex cells. So what could possibly come from this sex cell, from this diploid nucleus? And that's a big T and a little t, and again, a big T and a little t, because that's all that's there. And then we just play a weird game of tic-tac-toe, and we match. Big T, big T, big T, little t, big T, little t, little t, little t. So what we find is, if this family had 4 kids, 75% of them would be tall, because there's a capital T available, all right? And 25% would be short, because only 1 out of 4 is little t, little t. So that's dominant and recessive for heterozygous mixing. If we did homozygous mixing, so capital T, little t, same process. Break it, split it up, so you know which letters are which, and then put them into a table. And what you'll find is, if this tall person bred with a dwarf, all their kids will be tall. But if their kids were to breed with each other, they would have the same issue as this one. Okay, so that's monohybrid inheritance. What's this? Oh, then we have one more mix, homozygous with homozygous. Homo means two of the same, so little t, with heterozygous. And what you'll find is that that one's going to be 50-50. Okay, then you get codominance. This is where it gets fun. Now, codominance is when there are multiple alleles. Okay, there's multiple options, but you always have one that's dominant. So, and you also have one that's recessive. In our situation, IO is recessive. So if you mix IA with IO, you get AO, and what this means is that you're an A. If you mix A with A, you get AA, and what that means is that you're an A. If you mix B with O, you're BO, but the moral of the story is you're a B. You mix B with B, you're BB, moral of the story is you're a B. If you mix A with B, they both win. They're both dominant. So you get codominance, AB, and of course if you mix O with O, they're both recessive, so you get that. So the dark red are the dominant ones. They will always show if they're present. If they are in a room with another dominant one, then they both show. So imagine a classroom with 2 sets of loud kids. So student A is loud and student B is loud. If you put student A in a room with student O who's quiet, you're going to hear A. If you put them A with A, you're going to hear A. Does that make sense now? But if you put A and B, who are both loud, in the room, you're going to hear both of them. They don't drown each other out. It's just amplified. Lovely. So this is about blood types. You will get that example as it relates to blood types, and you have to talk about that. Okay? And I lost the mouse. So now we get into variation, and variation is differences within a species. Okay? So there are 2 types. You have continuous variation and discontinuous variation. And continuous variation, there are your properties. And basically an example of continuous variation would be height. You can, within our species, there are people at the lowest end, there are people at the highest end, and they're all over the place. All right? You can be 1 foot 5 inches, or 1 foot 6 inches, or 1 foot 7, or 1 foot 7.5. It can break down to anything. Discontinuous variation, you have set groups, and the example they love to use for that one is blood type. You're either A, O, B, or AB. There is no in between. You're in a group or you don't exist. Okay? And what you find is that this one is due to genetics, so discontinuous variation. It's either in your genes or it isn't. And then continuous variation is due to environment. Your height can change based on all sorts of things. So the examples they love to give are height and blood type. You could come up with any more on your own as well. The rolling your tongue and the type of earlobes is another discontinuous that they like to use. But there you go. So please be aware that your environment and what you've inherited differ in how you express. So yes, you might have the genes to be tall, but if you didn't sleep enough, eat enough, things of that nature, you won't be tall. So that's inheritance. Let's double check, see what the syllabus is saying. Continuous, two extremes, bang. Alright, next we move on to, well, there's your definitions, right? What? I had more work on that? Darn it. Alright, then we need to get into mutation and natural selection. So natural selection is, it's a form of mutation, or it's a mechanism for mutation, and the species will eventually change if certain things continue to happen. So in this situation, let's say we have a group of ducks. The slow, stupid ducks will continue to get eaten by the dog. Eventually that would only leave behind the fast, not so stupid ducks, and they will breed with each other, and that species would slowly change, and no more of slow, stupid ducks will ever exist because their genes will change. Alright? So that's the natural selection. We also have artificial selection, which is where humans do this. We find plants that have natural differences. So they all came from wild mustard, but when they started to make white flowers, we started to breed just the white ones, and then we get cauliflower. When they didn't have any flowers at all, and they just had buds, we continued to breed the ones that just have buds, and we got broccoli, etc., etc., etc. So they all came from one ancestor, but we artificially selected each type to get a different type of vegetable. Another vegetable example of that is carrots. Carrots are actually purple, not orange. We used artificial selection, and we ended up with a bunch of orange ones, and that's the dominant carrot that you see today, orange instead of purple. Alright, so we get into mutations, and mutations is a change in a gene or a chromosome. So let's think back to the genes, so the outfits. Alright, if I made a change in my outfit, I've made a mutation. So what should have been a blue shirt is now it's so fluffy. Okay, you can see how mutations can have a negative effect. So the one they want us to study is Down syndrome. And Down syndrome is when chromosome number 21 has 3 instead of 2. And this is called trisomy. Okay? And the properties of Down syndrome are here. And Down syndrome is one of the few trisomies where the organism can actually survive and live. Most trisomy results in people dying. Okay? Albinism is another mutation, not so much on the chromosomal level, but there you go. And then we have to get into, oh, that's just a joke, more mutations, sickle cell anemia and malaria. Alright, so sickle cell is a disease in which your blood cells change shape, and they look like that, and they struggle to carry oxygen, okay? And you can die from it. But sickle cell has a bunch of different options. You can either have all regular blood cells, which is a normal person, all sickle cell blood cells, which is extreme sickle cell, or you can be born with the 50-50. Some of your cells are like this, and some of your cells are regular. Now, the reason why it connects to malaria is mosquitoes carry malaria, and malaria cannot infect the sickle cell red blood cell, or it doesn't infect. So the people who have 100% sickle cell normally die, but the ones who have half sickle cell, half sickle cell, half human, they are resistant to malaria. So you'll find that in places that have lots of malaria, you also have a very high amount of people who are carrying the recessive sickle cell. Okay? So it's codominant. There's a parasite that goes from the mosquito into red blood cells that do not infect sickle cell red blood cells. So if you are heterozygous or 50-50, you're resistant. If you're homozygous and have all 100% sickle cell, you'll probably die. And if you're 100% regular, the parasite will infect you. So just for clarity, let's go back. A chromosome mutation. Down syndrome is a chromosome mutation. Albinism, which is none of these pictures, animals that are 100% white, that's a gene mutation. Okay, so sickle cell was is a mutation in an allele, so it's a gene mutation that causes the production of red blood cells to go awry. So now we get into superbugs, and superbugs are the form of, or they come from artificial or even natural selection, but we kind of force the hand. Let's say we have a germ, and germs reproduce very quickly, and because they reproduce so quickly, that means there are more chances for mutation. So in a regular petri dish, my germs will reproduce, and so will the mutants. So one of them, when my germ is reproducing, one of them comes out who does not die from hand sanitizer. So he starts reproducing, and has 1 billion children. One of his kids does not die from hand sanitizer. If I turn around and add hand sanitizer, all of his children die except for the one who was the mutant. So what does that mean? All the food that was once feeding the group of 1 billion is now there just for him. He eats that food and he multiplies. And now we have 1 billion of these guys who do not die from hand sanitizer. And there you have a superbug. Okay? Now instead of hand sanitizer, what we're actually talking about are antibiotics and medicine. The reason why you shouldn't use antibiotics so freely is because it increases the chances of making one of these guys. Okay? Genetic engineering is where we take the gene from one species and put it into another. So literally it's like having a leg of, not a leg, let's say we had, what are these things called? Cue cards, so you're a teleprompter, okay? So the show should go like this. The person's reading the teleprompter, it should say, "Welcome to the show. Tonight we are going to interview the President of the United States, Barack Obama." What we are able to do now is shuffle and change the genes. We can add something else in between. So now, when that organism tries to express this gene, it goes, "Welcome to the show. Tonight we are going to cook the President of the United States, Barack Obama." That changes the outcome. And the organisms that we like to use are called bacterial plasmids. They are circular rings of DNA or a code of DNA, and we're able to chop and slice and add whatever we want. So we use genetic engineering to make insulin. We find the human insulin gene, we give it to some bacteria, we add some sugar and some water, and the bacteria go crazy, and they continue to produce insulin whenever they're happy, and we just scoop the insulin off the top, purify it a bit, and give it to diabetic people. The reason why that's better than the other option is we sometimes can get insulin from cows and pigs, but there are some people with religious beliefs where they cannot use it from a cow or a pig. So I guess bacteria is the better option.